Hello, good morning, everyone. This is the extra lesson that we are doing in place of Mondays, and it is going to be on lecture 3, which is about the cell. So let's get right to it. Let me give you some introductory background information on cells. Now, we are familiar with this hierarchy, this organization from lecture 1. We talk about how complexity and organization being one of the characteristics of living things. And we know at the very bottom, there are things like atoms, like carbon, hydrogen, oxygen, nitrogen, phosphorus, that could come together to form important molecules in the body. And some molecules are small, like H2O, only 3 atoms. But in lecture 2, we learned about much bigger molecules, like macromolecules, such as carbohydrates, proteins, nucleic acids, and lipids. They are, thousands of atoms big, and we need them for various normal body functions. So now we're going to continue to move up the ladder. The next thing in this hierarchy of organization is cell and organelles. Right? So organelles are literally, they mean tiny organs that are inside the cells. And there are many different types of organelles. Each will have a specific function that allows the cell to be carrying out its normal job and being able to essentially be a living entity. Okay, and then the cell is the basic building blocks of living things in general. So we're gonna stay here for pretty much the rest of the course until lecture 8 and 9. Right? We're going to be learning a lot about what's going on inside the cell. We're going to look at how cell divides. We're going to look at what happens if that division process is malfunctioning. We're going to look at ways we can harness the various things that are going on inside the cell to our advantage. Like how do we use those mechanisms to produce medicine? How do we use those machineries to create better crops in agriculture? So we're going to be staying at this level for quite some time in the course. Now, it's hard to visualize how big a cell is. So, you will be doing your microscopy lab later on in the course, and you will be able to take a look at some cells in real life. But just to give you an idea, right? A human, like an adult, is about 1.5 meters, 1.5, 1.7, right? Depending on how tall you are. But that's a meter. Everybody is comfortable with what a meter is. It's like about this much, right? Okay. Now, if you shrink the size to about 100 millimeter, right? Then that would be like the size of a regular egg. And then if you shrink it again by 100 to 1 millimeter, 1 millimeter is the spacing between two black lines on a ruler, right? So that's about the size of a frog egg. Human eggs are much smaller. You can see it with a naked eye. And if we go down again by another 100, you see the unit is micrometer now, right? Before I told you 1 meter has 10 to the 6 micrometer. Right? So this is like a millionth of a meter is a micrometer. And animal cells and plant cells, they are in the range of 10 to 100 micrometer. Organelles are even smaller. They are in the order of 1 micrometers. Bacteria are about the same size. And when we talk about viruses, they are even smaller than bacteria. Viruses, they are measured in nanometers. 1 meter has 10 to the 9 nanometers. So a billionth of a meter is a nanometer. And when we talk about things like atoms, right, they are only 0.1 of a nanometer. Right? So this kind of gives you a rough idea of where things are in terms of size relative to one another. So these things we can pretty much see with our naked eye, but for these things we need to have what's called a light microscope. And then for things that are even smaller, to get really good details, we need to have electron microscope. I'll show you some of these things in a bit. So what is a cell? Right? A cell is defined as the smallest unit that has the properties of life. We already learned the properties or characteristics of life, right? Complexity, order, able to maintain homeostasis, energy requirements, so on and so forth, right? So all these properties are still being retained by a cell. Right? In fact, this cell is the smallest thing that has all these properties. If you go further into a cell, like if you look at the organelles that's within the cell, the organelle is not going to have that capability. Right? It's not alive anymore, right? You can think of the cell as the smallest unit of living thing. Now, we have various cells here. This over here is a bacteria. Okay, each ball is it's a bacterium. Some bacteria sometimes a ball shape, we call them coccus. Sometimes they are rod shape, we call them bacillus. There are different kinds. You probably have heard of streptococcus, right? Okay, people call it strep. When you get strep throat, you are being infected by streptococcus. Okay, so that's a bacteria. This thing over here, that's an amoeba. Okay, amoeba is they live in fresh water, like ponds or lakes, and they are a single cellular organism. It's just one cell base. Anybody know what this might be? This picture over here? It's in our body somewhere. You might have seen this before. What do you think this is? Feel free to type it in the chat or unmute, up to you, whatever you're comfortable. What do you think this is?
Is it septics?
Septics. It's a part of the body, though. Okay, which part of the body do you think this is?
Is it the nerves?
Yes, exactly. These are neurons. Very good. Okay, neurons in the brain, right? Okay, and they're making connection with each other. So my point of showing you this slide is that cells look very different, just like life in general looks very different, right? Remember in the first lecture I showed you pictures of like the jellyfish and the frogs, right? They all look very different, yet they have the same characteristics, the 7 characteristics that we talk about. It's similar situation here with the cells. The cells can look very diverse, but there are some core features that are shared by all cells, and they're listed on the next slide. These are common features of all cells. Doesn't matter if you're talking about bacterial cell, a human cell, a plant cell, they all have these things. Number one, they all have a cell membrane. The cell membrane, we briefly talked about a cell membrane is made up of the phospholipids, right? If you remember from the lecture 2. But cell membrane is essentially a barrier that separates the living from the non-living. Okay. Inside the cell membrane, it's alive. Outside the cell membrane, it's not alive. It's the cell environment, right? We can also call the cell membrane the plasma membrane. They mean the same thing. Secondly, all cells have genetic materials. All cells have genetic material in the form of DNA, right? In the form of DNA. And these DNA, as you learned from me last class, they contain instruction to make the various proteins for the cell. And finally, we have something called cytoplasm. So if I have to draw a cell, like a very primitive generic cell, right? It doesn't have to be a circle shape, but here we go. That would be the cell membrane. The liquid, the fluid inside, that would be the cytoplasm. and then there will be some genetic material. Okay, so these are the three key things.
I have a question, Professor.
Mm-hmm.
Can you please explain when you say cell membrane is a barrier between living from non-living? What is living and what is non-living we are talking about right here?
Yeah, sure. So like the cell is alive, right? So where does the cell end, basically, right? It essentially ends at the cell membrane. So the cell membrane contains the cell, which is like living, which is alive, and everything outside of that is non-living. Okay, so like for example, I can have a cell here and then a cell here, right? This is alive, living, and then in between it's just like fluid, maybe some water, maybe some dissolved minerals and whatnot. That's like the non-living environment.
Okay, thank you so much.
Yep, no problem. All right, so there is this something called the cell theory. It's a very important theory when people first discover cells. Okay, and you know, cell was mostly unknown to people until the invention of the microscope, right? Which we're going to take a look in a bit. But once they discovered the cells, then people started to observe them more, and eventually they came to forming the cell theory. There are 4 parts to the cell theory. Okay, number 1. Every living organism consists of 1 or more cells. Okay, the minimum number of cells you need to have to be a living thing is 1 cell. And there are many examples of organisms like that. Okay, this guy is called Paramecium. You will see it come up a few times in the course. Okay, Paramecium. It's a single cell organism, only 1 cell big. And just like amoeba, it lives in fresh water. Okay, if you take some fresh water from the nearby pond or creek or river and you look at it, you will find some of these guys. All right, this is C. elegans. Okay, C. elegans is a model organism. Model organism. What is a model organism? A model organism is an organism that that scientists knows a lot about. They know everything about them. Okay, down to what gene does what each part of a DNA does what. Okay, and we know a lot about them because we use them for research, and we use these model organisms to study the effects of new medications. Okay, to study gene therapy, to study many things. Okay, so C. elegans is a type of like a worm that that they use. Okay. Other model organism includes like a mouse, for example, right? So C. elegans, it's microscopic. You can't really see them with naked eye. It has about 3,000 cells in it. And when it comes to humans, we have trillions of cells. Okay, trillions of cells, 37.2 trillions of cells. This is, of course, just an estimation. Right? So the point is, first part of the cell theory, whether you're big or small, it doesn't matter. You are always going to be made up of cells. If you are living, you are made up of cells. At least 1, but can be many. Okay? Part 2. A cell is the smallest unit of life, individually alive, even as part of a multicellular organism. Okay? The cells in your body, they themselves are alive. In fact, if we take the cells from your body, we can grow them in dishes. These are Petri dish. Petri, okay. Petri dish. And the red stuff that you see here, those are nutrients, okay, nutrient solutions. What happens is we can take the cells out of your body, we can put them in one of these dishes, and then we can feed them with nutrients, and they will be able to live on their own. I mean, not forever, but they can live on their own for quite some time, as long as we supply them with the right nutrients. Okay, so we are alive as an individual, but on a cellular level, each cell is also alive on their own. Now, there was this
Sorry to
person.
interrupt, Professor. I'm so sorry.
No worries.
So, like in this, you said cell is individually alive. It's just a question. So, just say if we are not alive,
Mhm.
do you mean cell will be alive even for a bit, but would it be alive?
Oh, like if
Right.
you mean
Like
if
if
we
we
die,
die.
when we die, yeah,
Yeah.
When we die, the cells don't die right away. Okay, but they cannot live on for too long. Okay, because when we die, we don't breathe anymore, right? So without oxygen, they die. Okay, but there is a window. There's a window after a person dies where the cells can still be alive for a bit. That's why people have to be quick if they want to donate the organs after they die, right? The medical the doctor, they have to take out the organs quickly, put it on ice, right, and make sure to keep it fresh, right? So that's that.
Okay, thank you.
Yep, good question. Now, there was this person, her name was Henrietta Lacks, okay? Henrietta Lacks. And she had cancer. She had, I think it was ovarian cancer, a cervical cancer. Sorry, she had cervical cancer. Okay, cervical cancer. And what happened was the doctor treating her at the time took a bit of her cancer cells and was studying it. Okay, and then they found out, the doctors found out that unlike regular cells, these cancer cells don't die in the petri dish. Okay, as long as you feed them nutrients, they can divide and divide and divide and seemingly able to live forever. Okay, and that was how the first cell line was created. Okay, so it's named after the lady, right? Henrietta Lacks, Lacks. Okay, Henrietta. So this is the first human cell line. Okay, a cell line is something we can buy online actually, like from biotech company. They are immortalized cells. Sometimes they are cancer cells of origin. Other times we can do things to regular cells to make them immortal, and you can grow them like forever, right? And that's what they look like under the microscope. Again, I gotta emphasize the point. Cells don't have colors. Okay, they are very thin. They are transparent. When you look at microscope picture of cells and they're colored, it's because we dye them, right? For lack of better word. Like if you're really interested in how they do that, you can talk to me in person during break or something. But we dye them, right? So these are not the natural colors. We dye them. So HeLa cells are very, very useful. Right? Scientists have been using HeLa cells to study things like creating vaccines for polios, studying TB, Ebola, HIV, cancer treatment. Right? It's a very, very common cell line, and you can buy them online. Right? They're not cheap. For example, here's a 1 vial of it. Okay. And it's gonna cost you $740 Canadian. Right? But here's the thing: these are cell lines. You buy them, you can grow them. You grow them, and then you can put them into little vials and freeze them yourself. Right? You freeze them at -80, and then when you need more, you just unfreeze them and grow more. Right? That's why they're so expensive, because you only need to buy once, right? Okay, unless you kill them, and then you have to get new ones. Okay, so the Henrietta, she was never asked about whether they could use her cells for this purpose. Okay, so after she died, her estate actually sues the company that was selling these cell line. Okay, the company is Thermo Fisher. And they're saying, you didn't have her consent, and then you guys are like, making profits off her cells essentially, and then the family's not getting anything. Like, this is not right? It's not ethical. And so, that's the lawsuit. And then Fisher tried to counter-sue them, and then eventually, I think they settle. Okay, they settle for like I think a couple $1 million or something like that. Okay, so that's an interesting story to follow. But HeLa cells, unquestionable, it's very, very vital research tool. Why were we talking about it? Well, we're just talking about how, the second part of the cell theory says that the cells are individually alive, right? Okay? Normal cells cannot live forever, even if you give them the best condition. Okay? If you feed them nutrients and everything, they will eventually die. But cancer cells, on the other hand, okay, it could live outside of the person indefinitely. Why is that the case? Well, that's something you will learn later on in the course. All right, third part and the fourth part of the cell theory. All living cells come from the division of pre-existing cells. Okay, no one really knows where the first cell came from. I mean, there are theories about it. Okay, but we all know new cells have to come from the division of pre-existing cells. So here we are looking at onion cells, and you can see the onion cells is actually in the middle of dividing. So afterwards, you know, it will split, and you will have two new cells like that. Number 4, a cell contains hereditary material. These hereditary material is, you know, as far as we know so far, is all DNA. And these DNA needs to be passed on to the future generation. Okay, because DNA is, many people say, the blueprint of life. Okay, so those are the 4 parts of the cell theory. Studying cells. To study cells, biologists use microscope. Right? We learn about how our senses are kind of limited, and so sometimes we use tools to extend our senses to help with our observation. So the person who's credited with creating the first microscope is this Dutch scientist. He wasn't actually a scientist originally. He's like a tailor, okay? The person who makes clothes for people. He's like, you know, doing this microscope thing on the side as like an interest, hobby kind of thing. Okay, so he created this first handheld microscope. His name is Antony van Leeuwenhoek. All right. So in the 1670s, he created this microscope, and he was just observing waters from like the gutters. You know, like the house, they collect rainwater in the gutter. And he was just getting a sample and looking at it, and he was shocked to see there's like a completely new world in the water that he didn't know exist. Like you look at a drop of water, you don't think much about it. But when you look at it under a microscope, right, he saw all these things moving around and all these life, right? So he called them animalcules, okay? Like molecules with the word animal in front, right? But he didn't know that is actually universal, right, for life. Right? What he was observing was actually cells. And cells, as we know now, they are the building block for life, right? So on the right here, we have what's called a light microscope. That's the one you will be using in the classroom, okay, in your lab number 2. The light microscope, they are not too expensive. We got a good deal, actually. We were able to buy them at around $300 per piece. Okay, that might sound like a lot of money, but actually for microscope is not a lot. Now, wait until you see the next type of microscope. Okay, so for very, very small objects that you need to get a lot of detail from, things that are really small in micrometers range, then you really need to have high power microscopes. Okay, so we have 2 kinds here. We have what's called SEM, scanning electron microscope. You don't have to know the name, okay? But these are electron microscope. Instead of using light, they use electron beams to look at the specimen. And then we have transmission electron microscope. The difference is that the scanning can give you a 3D view, and then the transmission gives you like a cross-section view. And these microscopes are like really, really big. They can magnify up to 2.5 million times. Okay, you can zoom in 2.5 million times. Right? Whereas a light microscope, the ones we have in the lab, they are only good for 400 times. 400 times. So these can allow you to see like much closer with much more detail. These things are expensive, okay? Base cost of these microscope is about 1.5 million. And then you can buy like extra package, like, you know, softwares and stuff like that. Easily, it could be like 2-3 millions at the end. Right? So yeah, relatively speaking, $300 is not too expensive, right? Okay, done. One of the advantages of doing this online is I can launch a poll for you. Okay, so no more 1, 2, 3, 4 fingers. You can choose your answer, and it's anonymous. Okay, so take a look at this and let me know. Are you guys not able to see the poll or something? I only have okay, it's coming in now.
I cannot.
Okay, that's okay if you can't see if you cannot see the poll, just you know, do it in your head. All right, here we go. So, which of the following is a concept of cell theory? Well, we have D right here. A cell is the basic structural and functional unit of a living organism. Okay. Doesn't matter what kind of living organism it is, the building block for it is a cell. Next one. Okay. Earlier, I was telling you there doesn't matter what the cell look like. There are 3 things that are always there. Which one is not one of those 3 things? Okay, anyone else before I close the poll? Okay, excellent. Well, we drew it earlier. Let's see. Okay. There's the cell membrane, there's the cytoplasm, and then there's the genetic material, right? So the those are the things that are always there for all cells. Nucleus is not. You might not know what a nucleus is, but that's okay. I'm gonna show you. But you should still be able to do the question because you recognize, oh, cytoplasm, genetic material, and plasma membrane, right? So process of elimination. Nucleus is not present in all cells. All right, moving on. Let's take a closer look at the cell membrane and the nucleus. Okay, cell membrane and the nucleus. Now, very briefly, cells can be categorized into 2 groups. Okay, we can have what's called eukaryotes and prokaryotes. You can also say eukaryotic cells and prokaryotic cells. Mean the same thing. Okay, eukaryotes or eukaryotic cells. Prokaryotes or prokaryotic cells. So let's start with eukaryotes. We are made up of eukaryotic cells. Okay? They tend to be more complex. They evolved later in the lifetime of living things. Okay, the Earth first started with prokaryotic cells, and then later came the eukaryotic cells. It's generally bigger, and it has a nucleus. Okay, a nucleus. I'll tell you more about it in a second, but it's basically a special sphere that contains the genetic material. So you can see over here, there is the nucleus in the middle. And inside the nucleus, that's where you will find the DNA. Okay, so we have that. Our cells have that. The eukaryotic cells also have many types of organelles with different functions.
I have a
Okay.
question, Professor.
Yeah, go ahead.
So you said a nucleus contains genetic material, and in the previous slide we just discussed about what contains a cell. So it's a plasma membrane, and then there is genetic material. So basically, cell contains nucleus.
Okay, yeah. So the genetic material in a cell is always there, but it might not always be in the nucleus. Some cells have genetic material, but they don't have a nucleus. Okay, so like we have a nucleus to kind of protect the genetic material. For some other cells, their genetic material is just like flying around. There's no special chambers to put them in, no special sphere to put them in. Does that make sense?
Okay, perfect. Thank you.
Yeah. So there are many specialized organelles. Remember, organelles literally means little organ. They have different functions. Yes, go ahead, Nav.
Sorry to interrupt. So just following like up on the Bharti's question.
Mhm.
So does humans have different types of cells?
So all of our cells are considered as eukaryotic cells.
Okay.
Okay, but we do have different cells. Like a brain cell is gonna look very different from like a liver cell. Okay, but both of them are eukaryotic cells. Both of them are gonna have nucleus. Make sense?
Okay. Got it. Yes. Thank you.
Excellent. Okay, so what was it? The organelles, the little organs. Okay, there are many different types, and we're gonna learn all of them. And they have different functions. Okay, so there are many different types for eukaryotic cells. So animal cells, plant cells are examples of eukaryotic cells. Okay. Other examples like fungus. All right. Things like the amoeba, the paramecium. Okay, these actually belongs to some like a group called the protist. Okay, so these are all eukaryotic cells. Okay, all eukaryotic cells. On the other hand, the opposite of eukaryotic cells, we call them prokaryotes. Prokaryotes or prokaryotic cells. You can remember, P is for primitive. Primitive. Okay, they are very simple. They are smaller. They do not have a nucleus, and they only have a few organelles. Okay, just enough to keep them alive, I suppose. Bacteria are prokaryotes. Bacteria are prokaryotes. Does anybody know the name of any bacteria? Something that you've heard of on the news, something you read about. Usually there's like a single letter followed by a name. There's a lot of recalls for like salad and stuff like that because they're contaminated with whatever. I need a name. It's better if you tell me.
E. coli?
Yes,
Bacteria?
that's right. There's the E. coli, right? Okay. E stands for something that nobody can pronounce. So we shorten it to E. coli. Right? Does anybody have another one? Shauna, you have another one? Or did you say E. coli?
Yeah, I said E. coli.
Okay, good. Zoe, you have another one?
Listeria?
Listeria, that's right. Listeria causes listeriosis. Okay, so like all these bacteria, Salmonella, right? Anthrax, right? All these things are bacteria that are very simple, very small. They don't have a nucleus, so we call them prokaryotes.
Is Semolina was the one that because of which we recall the lettuce?
Yes, Salmonella is almost always Salmonella or E. coli when it comes to recall. Okay, Listeria sometimes make the news as well. Yeah, but usually it's E. coli and Salmonella. They recently just had a recall for what was it? Anyway, it's always on the news. There's so many I forgot. But there's always something. Right? It's because how they grow the lettuce. They grow the lettuce next to like animal farms. So sometimes the animal feces get cross-contaminated to the lettuce, and that's how it gets in the food chain, right? Yes, Yasmin.
Maybe we can give an example as an example bacteria of intestine.
Yeah, so you have a lot of bacteria in your intestine as well.
Intestine, yeah.
Yeah, but those are good bacteria, but nevertheless they are also prokaryotes. Very good. Yeah.
Okay.
So that's it. Eukaryote, more complex, bigger, have the nucleus. Prokaryotes don't. Let's take a look at the cell membrane. I'm just going to give you a very brief description of the cell membrane. Lecture 4, it's all about the cell membrane. By then, we will learn a lot more details about it. But for now, the cell membrane, that's the barrier that separates the inside of the cell with the outside of the cell. Okay? And the cell membrane is what we call selectively permeable. Okay? Selectively permeable. That means it allows some things to pass through and not others. That's what selectively permeable means. It allows some, but not all, things to pass through. Okay, which kind of makes sense. You don't want the cell membrane to be like a wall. If it's like a wall, nothing goes through, then how are we supposed to get nutrients into the cell? We also don't want a cell membrane that has no regulation. Right? If everything can pass through, then everything just kind of leaked out of the cell and the cell dies, right? And what about harmful things? We don't want harmful things to be coming in, right? So the cell membrane is selectively permeable. It allows certain things to come in while keeping others out. If it's bad for the cell, we try to keep them out. Is it a perfect system? No, not really. Sometimes harmful things do come in, but you know, it tries its best to keep bad things out. Okay? Selectively permeable. What is the cell membrane made up of? The cell membrane is made up of 2 layers of phospholipids, and you can see it over here, right? That is a phospholipid. You learned that from lecture 2, right? You studied that for the quiz. Phospholipid has a head, right? And that is a hydrophilic head. It loves water. It's able to bind to water. And then it has these hydrophobic tails. Right? So you're going to have 2 layers of these things. Right? That makes up kind of like a sandwich. Okay? The hydrophobic tails are all in the middle. And then the hydrophilic head is either facing the outside or facing the inside. And this would be like the inside of the cell, which we call cytoplasm, right? And this would be the outside of the cell. Okay, so I'm just going to pause for a moment for you to catch up and ask questions if you have any. All right, moving on.
Sorry, I have a question. So
Yeah.
outside of the cell, you said you mean like another cell or like
Yeah, I mean, it could be another cell, but it might not be. The best way to think about this is like if you think about a bowl of cereal, okay, you put like some cereal in, you know, like the little Cheerios or something. Like they are really close to each other, but then between them, there's the milk, right? So that's what it is in the body. Between cells is not empty, and it's not like completely stuck together with nothing in between. There is some fluid in between.
okay, it makes sense now.
Yeah,
Thank
so that's
you.
the outside. Yeah. All right, so here is another view of the plasma membrane. It's just like a two-dimensional view. You have the hydrophilic head facing the outside or the inside, both of which are mostly water, and then you have the hydrophobic tails in between. This is an actual picture of the real thing, right? Using the electron microscope, very high power, and this here, you see the two very dark lines. Right, the really dark lines, those are the hydrophilic heads. And then the little white gap in the middle, that's the hydrophobic tail. Okay, so this is outside. This is inside. Okay. Now, the plasma membrane is not just forming the cell membrane. Okay, it's not just the cell membrane. There are some organelles, as you will see, that are also bound by the same kind of plasma membrane. Okay, some of the organelles that we learn, you will see, have two of such membranes in some cases. All right, moving on. Now, I'm gonna draw this with you to help you understand it a little bit better. Okay, it usually is better if we draw it together. So please take out a piece of paper, and some color would be nice. Hey, do you guys say you have class in the afternoon, right? Like, is it? Did you say 12:30 or something?
12:30.
12:30. Okay, so I'll make sure I give you enough time to go to campus. All right, so let's take a look at this. All right. This is a cell. Okay, this is a cell, and then this is, what is this thing in the middle?
The nucleus?
That's right. Okay, so there's the nucleus. Okay. Only eukaryotes have nucleus, right? Plant cells do as well. Fungus, amoeba, paramecium. So what we're gonna do is I'm gonna take the nucleus and I'm gonna zoom in on it. Okay. Now, if this is your piece of paper, you should draw the cell here, and then you should zoom in here, and then I'm gonna keep on drawing stuff. Okay. Eventually, we'll write some notes here. Just planning ahead, right? Turn the paper sideways. All right. So here's the nucleus. Let's do this. Okay. It doesn't have to be pretty when you draw it. Just try to add some details to it. Okay. So there's the nucleus. And you know, I'm gonna cut open this nucleus a little bit. I'm gonna cut the nucleus open a little bit like that. Okay, just slice it open a little bit, and then we're gonna take a look at the inside in a moment. But for now, I want you to know that the nucleus is actually covered by a membrane.
Cell membrane.
Okay, that's actually a good thing that you mentioned that. Okay, the cell membrane is all the way out here, right? Like, remember, we're zooming it in. So this is cell membrane. Okay, cell membrane. And then this would be like outside, huh?
Mhm.
What do we call the inside, guys?
Nucleus.
This is the nucleus, yes. But what
Yes.
about like all this stuff inside the cell? What is this?
Cytoplasm.
Cytoplasm.
Yep, cytoplasm. Okay, I know it's a lot of terms. I'm sorry. I'm just the messenger. And you have to spend some time to go through the materials, right? Okay, here we go. I'm gonna color this guy. Okay, and I hope you have some colors with you, some highlighters maybe. Okay, so this here, this purple thing that wraps around the nucleus, this is called the nuclear envelope. Okay, nuclear envelope. Just stay with me for now. I'll give you an analogy at the end to help you visualize this. Okay, so what is the nuclear envelope? It is a membrane that surrounds the nucleus. Now, I want you to think about what we said. What is so important that is inside the nucleus? What do we have inside the nucleus? That is very
Genetic
important.
material.
That's right. We don't want to damage the genetic material. Remember last class I said if you change the genetic material, you might end up changing the protein,
Yeah.
like sickle cell anemia. It could cause you to become sick. So it makes perfect sense to have an envelope to surround the nucleus, right? So the genetic material cannot escape on its own, and bad things can go in to damage them. That's what the nuclear envelope is for. Now, sometimes things do need to go in and come out of the nucleus, but they are going to be doing it in a regulated manner. So on the surface of the nuclear envelope, you're going to have these little openings. Okay? Openings. These openings, they are called nuclear pores. Okay, so the nuclear pores allow materials to enter or leave the nucleus. All right, but not anything. It has to be like certain things. It has to be done in a regulated, controlled manner. All right. Any questions so far before I bombard you with more details? Excellent. Now, on the inside of the nucleus, and this is all real, right? Like I'm not making this up. On the inside, there is an other structure, another kind of like circular looking thing. Okay. So that is called the nucleolus. Nucleolus. If you need to read about these things, it's on the free online textbook. I'll post a link for you if you need to, but everything should be in the PowerPoint slides already. Okay, so this is the nucleolus, this guy. Okay, and the nucleolus, it's another structure that's inside the nucleus, and its job is to produce another organelle called ribosomes. Okay, but I cannot explain that right now at this point. We'll come back to it in a second. But just know that the nucleolus makes this other thing called a ribosome. So where are the genetic materials? Where are the genetic materials? Well, the genetic materials is going to be all here. Okay, all here. That's the genetic material. And we know it's DNA, but here is the thing. The DNA from a single cell, if you line it all up and you spread it out, it's very long. It's like 6-7 meters long. It's impossible to fit 6-7 meter long material into an organelle. Remember, organelle is a millionth of a meter big, right? So like, how do you pack it all in? Well, it turns out the solution is you need to wrap the DNA around some proteins. The best way to visualize this is, let me just show you the PowerPoint, and I'm gonna tell you how this is. Okay, so like this is naked DNA. DNA on its own is called naked. Okay, that's actually what it's called, naked DNA. Very long. Okay, all the DNA we talk about how there was 46 chromosomes, right? All the DNA in a single cell, it's like 6 meters long. You got to pack it all into the nucleus. How do you do that? Well, there are these special packaging proteins, which are shown here in yellow, and you wrap the DNA around them. Okay? It's like a yo-yo. You know the yo-yo, the thing, the toy, right? The yo-yo has a very long string, right? But you can wrap the string around the yo-yo. Right? Or you can think about like sewing thread, right? The threads you wrap around the spool, right? The core thing, right? So once you wrap them up, you can condense the DNA, and we call them chromatins. Right? So chromatin is just packaged DNA. When you have DNA that wraps around the proteins, they get more compact. You can fit them nicely into your cell. So genetic material is a DNA wraps around proteins to form chromatins. Okay. Chromatin. Okay, any questions so far? So let me give you a quick analogy to help you understand. I mean, you don't have to draw this next part. You can just watch me do it, I suppose. I think if you're confused, a good way to see this is to think of like an avocado. Okay, so here is an avocado. You cut it open. Okay, and then in the middle of the avocado, you have a seed, right? Okay, and then here you're gonna have the meat or something. Okay, so in this analogy, the seed is like the what?
The cell membrane.
Is that really okay? Good try. But,
Oh,
okay,
sorry,
this whole
sorry. The nucleus.
the whole thing is the
nucleus.
nucleus. That's right.
The nucleus.
Oh,
Yeah.
the nucleus.
That's right.
Yeah,
The nucleolus.
that one.
Yeah, that's the nucleolus.
Okay.
All right. So next time you have an avocado, you can think of it like this. And then where would the DNA be? Well, the DNA, the genetic material, will be like in the meat, right? Okay. And the skin of the avocado, that would be like the nuclear envelope, right? Is this working or not really? I don't know. I can't see your face. And then now if you take like a toothpick and then you poke a bunch of holes on the surface, then you have your nuclear pores, right? Is this a little bit better, maybe? Any questions?
Yeah, it is.
Okay, all right, good to know. All right, so that's pretty much it for now. All right, for the nucleus, I mean. So let's move on. Let's add extra detail to our picture now. Now that nuclear envelope that we have, okay, the nuclear envelope that we have that surrounds the cell, the nucleus, it turns out it doesn't stop there. Okay, after it wraps around the nucleus, it continues to branch out. Right? It will branch out like this and form these tunnels. Okay, tunnels. Okay, like that. And it will keep on going out, and then I'll change the color. They become like, you know, I mean, it's the same thing, but it's like further out, and there's a subtle difference in it. So I'm just gonna use a different color. All right, so I'm gonna zoom out a little bit to let you see what's going on. Don't forget the cell membrane continues like that. Okay. So this here is an extension of the nuclear envelope. Okay, as is this. Okay, it just extends, it spreads out, form a nice network in the cytoplasm. So these things, these extension, we have a name for them. Okay, they are called endoplasmic reticulum. Okay, so I'm going to put it here, endoplasmic reticulum. You just have to believe me. Reticulum means network. Endo is within. Plasmic, as in like inside the plasma membrane. So it's a network of membrane that's inside the cell. So that's endoplasmic reticulum here. We also have another endoplasmic reticulum here. Please write it twice. I will tell you why in a second. Now, some of these endoplasmic reticulum will have ribosomes on them. I haven't tell you what the ribosomes do yet, but we do know the ribosomes came from the nucleolus. Okay, so the nucleolus makes the ribosome. The ribosome will get out from the nucleus through the nuclear pores. Okay, through the nuclear pores. Right here, it will come out. The ribosome will come out through the nuclear pores, and some of them will attach onto the endoplasmic reticulum. Okay, so there's a lot of moving parts here. That's why I have to tell you a little bit at a time, okay? But they're all interrelated. So these dots, that's ribosomes. Okay. If the endoplasmic reticulum have ribosomes on them, then we call them rough endoplasmic reticulum, or RER for short. They appear to have these bumpy appearance when you look at them under the microscope. That's why we call them rough endoplasmic reticulum. Some endoplasmic reticulum do not have ribosomes on them, like these pink ones over here. So they're not rough. We call them smooth. Smooth ER. Smooth endoplasmic reticulum. Okay. Now, ribosomes do not necessarily have to be stuck on the ER. You can have ribosomes that are just floating around on their own. Okay, they're really small, they're really tiny. Okay, so that over here, that's the free floating ribosome. Okay, in cytoplasm. I'll post this note as well. Okay. Yes, question. Go ahead. Oops. Sorry, can you try again? There was a lot of feedback. Maybe your mic was too close to your speaker or something. One more time, please.
Can you hear me now?
No, not working. Can you type your question? If you don't mind. Okay, I'll answer them, okay? Just, yeah, okay, good. All right, anyone else have any other questions in the meantime?
I do.
Yeah,
So
go ahead.
the cell is composed of atoms, correct?
Well, yeah, but so are the organelles, right?
Yeah. So I mean, in the atom, there's also a nucleus.
Oh, yeah. Okay, you're not the first person to ask this. Yeah.
Okay.
Yeah, no, you're right. Yeah, the atom has a nucleus, but that's a different nucleus as like this nucleus. It's the same word but used in different contexts. Yeah.
Okay.
Yeah. Nucleus in general just means like a center, right? Yeah. So that's what it is. Yeah. Okay, let's see what Nav's question is. Can you tell me again what we call the rough pores? Like, are you talking about like these pores, or are you talking about these dots? Let me see. The dots. Okay, the dots, they are ribosomes. They are ribosomes. They are not pores. Okay, they are literally like little sesames, okay, if you would. Okay, they're dots. Yeah. Oh, I think Shauna tried to answer your question. Yeah. So like on the rough ER, on the rough endoplasmic reticulum, yeah, they have the ribosome. Ribosomes, R-I-B-O-S-O-M-E-S. Okay, ribosome. There you go. Is there
And
another?
just
Mhm.
oh, sorry.
Go ahead,
Just
Jefferson.
confirm the rough and the plasmic reticulum is called is called the rough and it's called the RER because it has the ribosome on it, and the SER doesn't have the ribosome and it's called smooth because it doesn't have the ribosomes on it, right?
That's right. That's right. Yeah. Yeah. It's just like
Okay,
an
okay, okay.
appearance. It looks bumpy, so we call them rough. That's all. Functionally,
Okay.
they're different though, okay? Which I'll explain in a bit. All right. So, you know, this is another way I like to tell students to study biology, right? Like don't just read the notes, right? Try to draw it out, try to write it out, try to construct it from scratch, right? When we construct it together like this and build it up from nothing, you know, it helps you remember it a little bit better. At least I hope it does. Okay, so we talked about this already. This is the professional drawing, right? You can see the nucleus, you can see the pores. The nucleolus is right here in the middle. We have the rough ER with all the dots. They use yellow for the ribosome, right? Same thing. And then further out, we have the smooth ER. Okay, but they are both an extension of the endoplasmic reticulum. Sorry, they are both an extension of the nuclear envelope. The nuclear envelope. All right, let's take a closer look at the ribosomes. Now I know they are really small, right? I told you like they're just the little dots when we drew it on our picture, right? The blue dots and whatnot, very small. But if you zoom in and take a look at the ribosomes, they are actually made up of two parts, right? Like a hamburger, right? Think of a burger, the burger bun. There's the top part, which is bigger, and then there's the small part, the bottom part, which is a little bit smaller. And that's what a ribosome is, okay? The ribosome, it's made up of proteins as well as RNA. If you take a look at the note we had on Wednesday, the PowerPoint slides, you don't have to do it now, but do it later. There are different kinds of RNA, right? We talk about mRNA, like the ones they use in the vaccine. And then there's another one called rRNA, right? That's the kind of RNA that makes up a ribosome, okay? But for now, you can just say it's made up of proteins and RNA. The function of ribosome is they make protein, okay? They literally connects the correct amino acid together to form a protein. Okay? Remember, the protein is made up of amino acids, you know, one connected to the other through the peptide bond. So the ribosome is the thing that actually connects them together. That's, you know, taking the carboxyl group and then the amino group, connect them together and keeps on connecting until you build the whole protein. This picture shows the rough ER. The little black, the black lines here, that's the endoplasmic reticulum, and all the like the, you know, it's not very clear, but I mean it's a limitation of the microscope, right? But all these little black dots here, all these dots, those are the ribosomes, right? Okay. Recap. Ribosomes are made from the nucleolus. They can be floating around on their own in the cytoplasm, or they could be stuck to endoplasmic reticulum, which makes it rough ER. What does ribosome do? They make proteins. They make proteins. This is literally what I just said. I forgot I have a slide on it. But look, some are freely floating, some are attached to the ER. All right. Now, there is a central theme in biology. Dogma is just like a special fancy word for theme, okay? They call it central dogma of biology, central theme in biology. And it is like this. We have DNA. Right? We have DNA, the blueprint of life. DNA, the ACTGs, right? Okay, so the ACTGs. The DNA contains instruction. Contains instruction to make protein. It's not new idea. I told you this before already. Contains instruction to make proteins. Now, DNA is very long. Okay, you can think of DNA as like, you know, an encyclopedia of cooking recipes. Right? Thousands and thousands of recipes in there. Right? If you want to make one of the recipes, and let's say they don't let you take the encyclopedia out of the library or something, what are you gonna do? You really like a recipe in there, but they don't let you take out the book, and you don't want to because it's so big.
You make a photocopy.
Exactly, you make a photocopy. Although nowadays people just want to kind of take a picture, right? Okay, but yeah, you're right. Okay, I was thinking about photocopy. Are you gonna photocopy the whole book? Absolutely not. You're just gonna photocopy the recipe that you need, right? And that's what we are doing. When we need to make a specific protein, we're not gonna drag the whole DNA out of the nucleus. That's not safe, and it's, you know, very bulky and it's very long, right? So we only copy the section that we need, right? In the form of messenger RNA. Okay, so messenger RNA is a copy of DNA. Right? Again, not the whole thing, just the instruction that you need to make a specific protein. We have many proteins in the body. Let's say you want to make insulin, then you find the instruction to do that, and then you will copy only that part. Okay, you copy.
Sorry, I have a question.
Mhm.
Sorry, I just got confused. So we were just talking about ribosomes and
Mhm.
how they help to connect amino acids to form protein, right?
Yeah.
So this is like when we are copying from DNA, it's like how we are connecting those amino acids.
Okay, hold on. Now, when I finish explaining this, hopefully it will answer your question. If not, ask
Okay.
me again, okay?
Okay.
So now we have the DNA, which is ACTGs, and then we make a copy of the section that we need in the form of RNA, which is ACUG, right? Okay, exactly how this happens, we will learn later in the course. But now we have a copy, we made a photocopy of the recipe, then we can take that home with us in our analogy, right? So we can take this mRNA and we can bring it out to the cytoplasm. In the cytoplasm, the ribosome now comes and essentially read the instruction on the messenger RNA. Okay, the instruction tells the ribosome which amino acid you need to connect together. And then the ribosome does it. Right? So the flow of information goes like this. DNA, and then it becomes RNA. This is called transcription. Okay, because it's the same language. DNA and RNA are the same language. They are both nucleotides. A, C, G, and then T or U, right? They're both nucleotides. Transcribing, you're copying. And then in the cytoplasm, the RNA is gonna be decoded by the ribosome, and then you're gonna use it to make protein. This is translation. Translation. Because RNA and proteins are different language. RNA is nucleotides, but proteins, they are amino acids. That's why it's called translation. Okay, so it happens in 2 stages. Does that answer your question, Bharti, or you have a follow-up?
Somewhat. I'm trying to figure it out.
Okay, so ask again the question. Let me try to address it.
Okay, so basically we are saying that ribosomes helps to make a protein,
Yes? Mhm.
right?
Yeah.
So if so from the messenger, so that RNA that ribosomes are getting, So ribosomes are making proteins, but how they're making it is basically that's what you're trying to say, that RNA is giving the instructions
Mhm.
so that how it's gonna make it. So ribosomes does not make protein itself. It needs to have instruction coming to it. That's what you said?
Yeah, basically, right? Like so the ribosome does make the protein, but it cannot do it on its own, right? It needs to have the recipe, right? It needs to have the instruction to do it, right? That's the best way to think
Okay.
about it.
Okay.
All right.
Yeah.
Yeah.
Thank you.
Okay. Everything else, everyone else okay? Any question? Okay, maybe we take a quick break, just like, 5 minutes, just for you to stretch your legs. If you think of anything else to ask, feel free to do that. We'll resume in a bit, okay? 9:45. Okay, we'll come back at 9:45 and we'll do more. Okay, thanks. All right, let's keep going. Now I want to show you this because it's a little bit abstract for some of us, right? To, like think about what exactly does all this mean. Now there's something called the genome browser. It's online. Anybody can access it. I'll paste the link in the chat box if you want to play around with it later. But this is, I search up insulin, okay, the insulin gene, and it's on chromosome 11, like right there. And what we can do is like, I can click into it and I can actually get the sequence, right? Like this is the result of something called the Human Genome Project, which started, a long time ago, like almost 20 years ago. Okay, and it has been completed since then, and we have the information of all the genes that are in our DNA, and so that's the instruction right here to make the insulin. It's just A T G G C C C blah 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 all the way over here, right? It's not a particularly long instruction. Some of them are much longer, right? And so of all the ACTGs in your entire DNA library, you only copy this part in the form of RNA. Okay? And so I think we have that option as well. Like we can look up the RNA. Do we have the RNA here? Okay, maybe not. Okay, but like, so you're gonna take that, you're gonna make the RNA, and then you're gonna be able to make the protein from it as well. Okay. Oh, right here. This is the RNA. See? So you make a copy of it. It looks much shorter, and there is a reason for it, which you will learn later on. So from the DNA, it becomes the RNA, and then from the RNA, you can make the protein. So the protein is this. Okay, each letter represents an amino acid. So for insulin, it is only 110 protein, 110 amino acids. Okay, so that's basically what happens. From DNA to RNA, we call that transcription, which happens in the nucleus. And then from RNA to protein, we call that translation. That happens in the cytoplasm with the help of ribosomes. All right, concept check quickly. Which of the following is a prokaryote? Let's launch the poll. Go ahead. Right, anyone else? Okay, for the interest of time, I'm going to stop there. Yeah, and everybody who answered got it right. E. coli is a bacteria. A bacteria is an example of a prokaryote. Okay, liver cell, that's in us, that's eukaryotic. HeLa cell is the cancer cells from humans, eukaryote. Amoeba, also eukaryote. How about this one? If you took the notes, you will see that the nucleolus up here, it produced another organelle called the ribosomes. Okay, so the correct answer is A, produces ribosomes. All right. So I am just going to finish up with the endomembrane system. Okay, it should not take too long, maybe 20 minutes or so, 20, 25 minutes, we'll see. But we'll definitely end no later than 10:15, okay? So endomembrane system. The endomembrane system, as the name implies, it's a network of membrane that's found inside the cell. It's in the cytoplasm and it's made up of a bunch of membranes. The system works together to produce, okay, synthesis means to produce, to process and digest various things, including lipids, proteins, and other large molecules. Okay, so they make them, they process them, and they even digest them. In some cases, they play a role in recycling and disposing of molecules and particles. Some things we can reuse, right? After you break down a protein, we have all these amino acids. Hey, let's not waste them. Let's use them to build other proteins that we need. In other cases, we don't have any more use of some things and maybe harmful to keep them in the cell, then we try to dispose of it. That's what the endomembrane system does. There are many different components involved in the endomembrane system, some of which you already know. Okay? For example, there is the nuclear envelope. There is the rough and smooth ER that we drew, but we didn't talk about what they actually do. And then there's something called the Golgi bodies, also known as the Golgi apparatus, and the lysosome. So let's take a look at the flow of information here. Okay, I'm just gonna draw it for you on the note, and then when we return next week, the first thing I do is I'll go through the PowerPoints with you to help you refresh your memory. Okay, I think that's probably the best way to do this. Okay, so let's go back to our lovely drawing right here. All right. So endomembrane system. We have the nuclear envelope. We have the rough ER. So what I'm gonna do is actually I'm gonna zoom in a little bit more and see what's going on with the smooth ER over here. Okay, and I'll post like a video for you to see later, like an animation, to show. I never like to show animation in class. I think it's a waste of time. It's like you can I mean you can watch it on your own, right? And you definitely should. But I'll try to explain this to you. So here is a ribosome. Okay, here is a like zoomed in version of what's going on. There's the ribosome. It's actually two parts, like the hamburger, right? And we're gonna have like the messenger RNA right here. The copy, the instruction, the recipe, right? To make the proteins. So maybe watch first and then copy, right? So you understand what's going on. The ribosome will read the instruction on the messenger RNA, and then it's gonna go ahead and build the protein. I'm just gonna use green to represent the protein. The protein is gonna be built directly in the rough ER. Okay, so there is your protein. Protein. Now, do you need to build the protein with the rough ER? No, not necessarily. These free floating ones, they can make proteins as well. It's not a problem. Okay, but in terms of the endomembrane system, I'm gonna describe it with the rough ER here. Now, once you make the protein, what happens is the protein will move towards the edge. And this is the part where it's a little bit difficult for me to show. But the protein will fold up. And then what's going to happen is, I'm going to draw the next picture over here. What's going to happen is, you gotta squeeze the end a little bit like that with the protein inside. Okay, and then moments later, it will pinch off. it will pinch off like that. Can you visualize that? The ribosome makes the protein goes into the rough ER, and then it moves towards the edge where it eventually bubbles off. It's like blowing a bubble, literally. Okay? And here we have a vesicle containing a protein that's freshly made by the ribosome. Questions? Okay. Again, if you got to go to campus, go. Okay, leave. Don't worry, I'll finish doing this recording and I'll post it online. Okay?
So, I'm sorry.
Yeah.
So you said that proteins are like, say, pushed to a corner and then it detaches itself?
Yeah, but with a bubble though, right? With the membrane. Okay, the membrane was part of the rough ER, right?
Rough ER.
Mhm. Okay, let me show
So
you.
basically, rough ER is detaching itself.
Yeah,
It's
in a
making
way.
a bubble, but it's detaching itself and
Mhm.
it's containing protein in it. So it just that's the bubble of protein we called it. But it's eventually it's the rough ER that it detached itself, but it detached itself with the bubble containing the protein.
That's right. So like in this
Okay.
picture, right? Yeah, it's gonna butt off
Okay.
into this little bubble. Okay. All right. So what's gonna happen to this bubble? Now, another part of the of the endomembrane system is that is a thing called like the Golgi body, and the Golgi body is basically like a stack of membranes like this, more like pancakes. Okay, one after the other. It's not connected like the rough ER, right? And so we call this the Golgi body. Okay, Golgi body. So the vesicle that we just got from the rough ER is gonna come over here to the Golgi body. And then the Golgi body will sort the protein. It might make some modifications. So it'll pass the protein forward and sometimes it'll pass it backwards, a little back and forth. And eventually the Golgi will repackage the protein in a new vesicle. There we go. And then the protein inside is like slightly different. So I'm going to use a slightly different green. Okay, let me write that down for you. The Golgi body. The function of the Golgi body is to sorts, modifies, and repackages protein. And you end up getting this thing. You end up getting this other vesicle here. Okay. So that's the story so far. You know what? I think people might need to go to campus. I see a lot of people leaving. So I think maybe this is a good place to stop. I think we got pretty caught up with the schedule. We should be able to wrap this up on Monday. But we will also start on lecture 4, right? So make sure you print that as well. Thank you so much for coming on this extra class. Good luck with your afternoon class, and I'll
Can I
see
ask
you
you a
all.
question?
Yeah, sure. Go ahead.
Sorry.
No
I
worries.
might be out of context, but so I'm just gonna go with the start. So ribosomes,
Mhm.
they formed proteins,
They do, yeah.
right? And they formed proteins based on the instructions from RNA, the
Yes,
messenger RNA that gave
correct.
it to it, right?
Mhm.
And then those proteins were pushed down by the membrane and then detached itself into a bubble.
Yep.
And now you said now Golgi
Golgi
body
body.
comes in. Yeah, Golgi body comes in, which is sorting and modifying and repackaging proteins. So what it is sorting and modifying it based on?
Yeah, that's a good question, right? So when you make all these proteins, like I haven't finished the story yet, but long story short, when all these proteins come in, like we need to know where they go. think of the Goji body as like the post office. You have all these mails coming in. You need to know which mail goes where, right? Does it go out of the country? Does it, go where, right? So that's what the sorting does, right? Where should this
Okay.
protein go?
So
And
that
then
instruction is just for the formation of proteins, not like it does not instruct how to sort it.
that's right? So sorting is based on something else that, we don't really have to
Okay.
know for this
Okay.
course. Yeah.
Okay.
And then, sometimes the things might need to be repackaged for like, if you need to ship this thing overseas or something, you got to make sure you tape it nicely. So that's kind of like what the repackaging does. Okay.
Okay, so the role of the messenger RNA ends up when ribosomes creates proteins. Their role
Exactly.
ends.
That's all it
Okay.
does. That's right.
Okay, perfect. Thank you. Sorry.
No worries. No worries. Yasmin, you have another question? I saw you unmute.
Yes, I have a question. It's out of topic, but I want to ask you.
Mhm.
Are you gonna repeat this class again?
No. This is it. Yeah. That's why we're doing it, because we don't have enough time to do it like in person, right? But I will post the recording though, so you can watch it again. Okay. Yeah.
Okay.
All right.
Okay. Thank you so much.
Feel free to email me, guys, if you have anything else you want to ask. Otherwise, I will see you all early on Monday. Okay. Have a good one. Thank you again. Bye-bye.
Thank you.
Yeah.
Thank you. Have a great day.
You too.
Thank
Thanks.
you.
Bye-bye. Bye, guys.
Bye.
Sir.